Welcome back to Newtonian, and in today's lesson what we're going to be covering is a chapter of biological molecules, and we're going to be covering starch. And this is part of the series on carbohydrates. So first of all, what is starch? Well, in previous lessons we've learned that cells, they have glucose in them, and which is then used in respiration. However, we also learned that glucose is osmotically active. In other words, we learned that glucose affects water potential. In other words, glucose within a cell can cause massive problems to the osmosis that's taking place. So, glucose is not the ideal storage method for a cell. So what's the storage method that a cell uses for energy? Because a cell eventually will need glucose. So is there something we can do to this glucose to store it? Well, there is. So the way we store glucose in cells, such as plants, is in the form of starch. Now, starch is specific to plants. And glucose storage in animal cells is done by glycogen. And that is for animals. So animal cells have glycogen, whereas plant cells have starch. Okay, so that's the first thing you need to understand. Now, the next thing you need to understand is a bit more about starch. Now, the next thing we understand about starch is what actually makes up the starch. Well, starch is made up of two things. The first thing that starch is made up of is amylose. The second thing that starch is made out of is amylopectin. Now, I know when you're looking at this, you're like, amylose, amylopectin? God, those sound like hard names. But to be honest, they're just fancy names for rather simple stuff. And that's something you'll come across in science a lot. So you're going to have to memorize that starch is made out of amylose, and it's made out of amylopectin. Yeah. That's kind of it about what starch is made out of. Now what we're going to do next is we're going to go over the structures of amylose and amylopectin one by one, and we're going to figure out why they're important, what are the properties that are important, and how this can come up in an exam question. Right, so now what I've done is I've drawn out the structure of amylose, right? So we can see this is a long chain of molecules. So this is a polymer. Now, the first thing we need to identify is what is the monomer? Well, the monomer here, actually, is something we've already covered. This structure here, this structure here, this structure here, I hope you've memorized, because these are all alpha glucose molecules. Right, so these are alpha glucose molecules. Now, look between each one of these alpha glucose molecules. So let's say this is alpha glucose molecule 2, this is alpha glucose molecule 3. Between these two, there is something a bit different. What is it? But what's different here is the presence of this oxygen here. And it's like a bridge. So we can kind of get a car that can cross this bridge, right? Can go zooming past from number 2 to number 3. So what is this called? An oxygen bridge. Now, an oxygen bridge is present between these two alpha glucose molecules, or between each of these alpha glucose molecules in amylose. But what is an oxygen bridge also called? An oxygen bridge is also known as a glycosidic bond, right? So it's also known as a glycosidic bond. So you've got alpha glucose molecules. Alpha glucose makes up our amylose, and it has glycosidic bonds between each other. Now the next important thing that you need to actually identify is how can we name this glycosidic bond? Right? If I want to specifically tell someone what type of glycosidic bond is present between this and this molecule in amylose, how can I do that? Well, scientists actually came up with, for once, a simple method of doing this that actually makes sense. Oh my god. So what we do is we look at this oxygen here and we always count clockwise, right? Clockwise. Now we start from this oxygen, we go clockwise, and each carbon we come across, we name that carbon as 1 and then so on. Okay, so this carbon here, we'll name this as carbon 1. This carbon here, we'll name as carbon 2. This carbon here, we'll name as carbon 3. This carbon here, we'll name as carbon 4. Same thing we'll do on this one. Okay, so from the oxygen we go clockwise. This is the first carbon here, so we name that carbon 1. This is the second carbon here, we name that carbon 2. This is the third carbon here, we name that carbon 3. This is the fourth carbon, we name that carbon 4. So now our car is going across the oxygen bridge from this alpha molecule to this alpha molecule. And it's going from carbon 1 to carbon 4. So how can we name this glycosidic bond? Well, it's easy. We just call it a 1, 4 glycosidic bond. So in amylose, you have alpha glucose molecules which each have a 1, 4 glycosidic bond present between one another, right? So that's kind of all of the structure of amylose. Now, one very important thing you need to note with amyloses is that it is not branched at all. Okay, wait, let me clear a bit of the board out and let's go over the properties of amylose. Okay, let's cover that. What else should we? Let's remove this here. Okay, and we'll write the properties. Okay. We can put that one there. We can do the numbering here as well, to be honest, just so that it sticks in your mind. 1, 2, 3, 4, and you can see 1 to 4. Okay, now let us go over the properties. Properties. What are the properties of amylose? Well, the first property of amylose that we have is amylose is insoluble. Amylose is insoluble. Now, why is this important for us? We recognize that glucose, which was polar and hence was soluble, meant that glucose affects the water potential of the cell, whereas starch is insoluble, so it does not affect the water potential of our cell. So this means does not affect the water potential of the cell, so is ideal for storage. So you can store this in as much large amounts as you want. It's not going to affect any of the water potential in our cell. So that's very, very good. Okay. The next property of amylose. The next property of amylose is that it's very compact. And why is it very compact? That's because amylose, this actual 1,4-glycosidic bond, it happens, in reality, it happens at a slight angle. So what that means is this slight angle will cause for a coiling nature to be formed, a helical nature to be formed. So our amylose is stored as a helix. Helix allows for compact storage. Hence, this allows for large amounts to be stored in the cell. Now, I want you guys to have a look at the structure. Okay, we've understood these two first points. What's a disadvantage of amylose? Why isn't amylose perfect? Why is it that we need amylopectin as well? What's the problem with amylose? Well, when I said amylose only has 1,4-glycosidic bonds, that means amylose is just one straight branch, which just keeps on going, keeps on going, keeps on going, right? It's just like a train. Now, I actually want you to stick with that train example, right? So you've got a lot of carriages, right? But on the fronts of each of the trains, you've got the place where the driver is, right? Depending on which direction the train is going. So you've got the driver on this side or on this side. And that means that the train can only be controlled from these 2 points, right? But also, that means if I want to remove some of these carriages from my train, I will first have to sever this point, and then I'll have to remove my carriages. And that means that there's only 2 real points that I can remove carriages from, which means we can't break this train thing down very quickly. Whereas imagine if I had carriages which were attached like this, like this. So I could break the carriages down from here, I could take them down from here, I could take them down from here, from here. There's a lot more points. And this is the point I want to hit home with amylose. Amylose only has 2 points, which is at the start and the end, from where we can break down amylose from. So enzymes only have 2 accessible sites in amylose. Because amylose only has 2 accessible sites, it's very bad for energy release. It's not good, which is why it makes it a very good long-term energy store. So unbranched, unbranched, hence good long-term energy store. So this is not good in the short term, it's good in the long term, which is why we have a combination of amylose and amylopectin. Right, so now we've gone over essentially what amylose is, what some of the properties are. To recap, the properties of amylose you need to remember is amylose is insoluble, it has a helical structure which allows for compact storage, and it's unbranched, which makes it a good long-term energy store within plants. And that's kind of all we needed to know about amylose. So now we're going to move on to the next one, which is amylopectin. Right, so now what I've done is I've drawn out the structure of amylopectin. Now from what you can see here, the structure of amylopectin is very similar to amylose. So you can see here that there is an oxygen here, and this is between the first carbon of one of the alpha glucose molecules and the 4th carbon of another alpha glucose molecule. In other words, you've got a 1,4-glycosidic bond still present here. Now, what's the difference? What's the difference between amylopectin and amylose? Well, to understand the difference, we need to number this alpha glucose molecule as well. So this will be number 1 carbon, number 2 carbon, number 3 carbon, number 4 carbon, number 5 carbon, and finally number 6 carbon. And we're gonna number this alpha glucose molecule as well. Same way, number 1 carbon, number 2, number 3, number 4, number 5, and finally number 6. So what we see very interestingly here is we've got an oxygen, the same way we had an oxygen bridge here, we also have an oxygen bridge here, which means that there is a glycosidic bond present between this alpha glucose molecule. We'll just name this to be number 1 and this one to be number 2 alpha glucose molecule. We can see that there's some sort of glycosidic bond present between each other. Now we saw between these two, we can name this number 3, between number 1 and number 3 alpha glucose molecule, we saw there was a 1,4 glycosidic bond present. However, when we look at alpha glucose molecule 1 and alpha glucose molecule 2, we see that the oxygen bridge is present between the first carbon of this alpha glucose molecule and the sixth carbon of this alpha glucose molecule. In other words, the glycosidic bond that's present here is no longer a 1,4 glycosidic bond. Instead, it is a 1,6 glycosidic bond. Now, why is this 1,6 glycosidic bond so important in the structure of amylopectin? Well, let's just see as an overhaul, what does this do? As an overall, what's it changing to the structure? So imagine this was just amylose, okay? And we have this just straight structure going on. And we look up, we're like, hang on, what in the world is that? What's this here? We see that there's another branch being formed here because we can see there'll be more 1,4 glycosidic bonds and more alpha glucose molecules on this side, right? So what we've actually done is we've joined two branches together. In other words, the 1,6 glycosidic bond allows for branching. It allows for branching to occur. In other words, amylopectin has branching in it. It is not dissimilar to amylose, where amylose does not have any branching. Now we're going to learn the significance of this branching in a second. So we've learned that our amylopectin has a 1,6 glycosidic bond between one branch and another branch, and this occurs approximately every 25 alpha glucose molecules. You don't need to by any means know that off by heart because it just varies, to be honest. So that's just an estimate or a rough figure, which is kind of meaningless. It's just to give you an overview. Now, a mistake that a lot of students make is they think that, okay, 1,6 glycosidic bond, we just memorize that and we're all good to go. And then they forget that amylopectin does have a 1,6 glycosidic bond, but it also still has 1,4 glycosidic bonds. So remember, amylopectin has 1,4 glycosidic bond as well as 1,6 glycosidic bond. So it has both. Whereas amylose only and only has a 1,4 glycosidic bond. Right, so we've understood the two types of glycosidic bonds that are present in amylopectin. Now let's go on to the properties of amylopectin. Now for that, what I'm going to do is I'm going to rub off this whole structure, because I don't think you can really understand or appreciate the properties of amylopectin by looking at a chemical structure of it. Instead, we need something different. We need something better. So let's do that. Who knew bio could be this fun? Rightio. So, let's draw amylopectin in a more useful way. Amylopectin. We've drawn it, we've written the word very big. Now, amylopectin, if we zoomed out, how would it look? Well, amylopectin would look somewhat like this. So you've got one branch there, you've got it attached there with another branch, and this branch is another branch, and that has another branch, this has another branch, this has another branch, this has another branch, this has another branch, that has another branch, that has another branch, this has another branch. Yeah, we could go on all day like this and might become a bit boring eventually. Yeah, but you get the concept, don't you? It's got a lot of branches, right? Compared to amylose, this guy is mega branched. Remember that. Amylopectin, mega branched. Amylose, boring, no branch. Yeah. So, why have I drawn this out like this? What am I trying to show you guys? Well, imagine if you're Mr. Enzyme, you're here. You're enzyme. And you need to break this apart. Okay? Now, compare this to amylose. Okay? So, enzyme comes along and he's like, okay, how am I going to do this? It realizes there's only two places it can take the subunits off from. Well, one place is here, and the other place is here. So, realistically, only two enzymes can actually work on amylose in one go, which is dead slow. Right. Instead, amylopectin's a bit of a smart dude. He's much better. Because he's got so many more accessible sites. You can have an enzyme working here, it can have an enzyme working here, and 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 here. So essentially, amylopectin can have a lot of enzymes working on it to break the thing apart into alpha glucose molecules very quickly. You can have a load of them. And if you have a load of them, this breaks down very, 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 very quickly. Whereas this guy breaks down very, 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 very slowly. So which one's better for energy release? If I want a lot of alpha glucose molecules in a very short time, do I want this guy or do I want this guy? Yeah, it's this guy, right? It's not even a thought. This can be broken down very quickly. So amylopectin is very good for that medium range energy supply because we said glucose is good for short term. Amylose is good for long term. What's good in the middle? Amylopectin. Right, amylopectin is good in the middle because it releases the monomer glucose which is required in respiration. And this is done rather quickly in plants using amylopectin. So you've got a combination of amylose and amylopectin present in your lovely plants. Now, if I'm going to summarize the properties of amylopectin, let me do it now. So, yeah, let's just put all of them here. So amylopectin, like amylose, is insoluble. That's insoluble. Number one, it's insoluble. Does not affect water potential. Woohoo. Yeah, okay. Number two. This one has a lot of branching, so it's very good for energy. Good energy source. Good, actually we'll call it good medium-term energy store. Medium-term energy store. Rightio. We've kind of covered all of amylopectin here, haven't we? Well, to summarize, amylopectin, whole lot of branching, insoluble, doesn't affect the water potential, is a good medium-term energy source. And that's all about amylopectin. The next guy we're going to be covering is glycogen. Now what's interesting about glycogen is it's not in plants. You see, animals require a whole load more energy. So we need something a bit more superior to plants. And that's why we've got glycogen. And that's the thing that's found in our muscles. And that's what allows us to contract them a whole load quickly. Let's go over that now. Okay, so now what I've drawn is I've drawn the structure of glycogen. Now you're going to look at the screen and you're going to be like, oh my god, what in the world is that? Because when I was an A-level student and I looked at this diagram and I was like, God, I don't understand a single thing on here. It's a nightmare if you think about it. It's a lot of chemical structures, and who likes looking at chemical structures? I personally don't. But trust me, this one is actually very simple. What you need to do when you're looking at one of these structures is you need to have a look at the individual ones. What's actually going on? What's familiar to me? Well, the first thing that should be familiar to you here is actually this. Each one of these rings is actually just an alpha glucose, right? This is alpha glucose, this is alpha glucose, this is. All of them are alpha glucose. What's the next thing I can do? I can start numbering them. This is carbon 1, this is carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. Ooh, let's do this one as well. 1, 2, 3, 4, 5, 6. What do I see? I see a 1,4 glycosidic bond. 1,4 glycosidic bond. What else do I see? Let's number this. 1, 2, 3, 4, 5, 6. Oh, I see that there's a 1,6 glycosidic bond. 1,6 glycosidic bond. Right? All we're doing is we're identifying things we've already seen. In other words, this diagram we've already seen actually. If you haven't recognized this, this is literally the same thing I drew before for amylopectin. But what's different here? The only thing that's different here is there's a whole load more branching. There's a lot of these branches, 1,6 glycosidic bonds. You've got one here, 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 you've got and here. You've got a lot of these 1,6 glycosidic bonds. What's going on? Well, what's going on is that this glycogen is very, 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 very branched. Okay, it's much more branched than amylopectin as well. And clearly it's more branched than amylose. Please. Yeah. Okay, so glycogen is extremely branched. Why is that beneficial? Well, glycogen being extremely branched is very beneficial because, as we discussed before, I'm just going to rub this horrible diagram off. So, yeah, it's scary, but it makes sense, okay? 1,4 glycosidic, 1,6 glycosidic. So remember, there's a lot of 1,6 glycosidic bonds here. Yeah, that's the point I'm trying to make. There's a lot of branching here. Rub this off. We don't need to look at that again. Guys, we're not studying chemistry. We're studying bio, okay? So even if you don't get these chemical structures that much in your head, it's fine. And for you guys out there who are trying to be medics and you've got chem as well, unfortunate. Well, yeah, unfortunate. That's all I can say. Anyways, so let me just draw this branched structure out again. Now, this branching is a lot more than what we had with amylopectin. Right, this is... You get the point, right? It's a lot more, right? A lot more branching than you saw in amylopectin. So glycogen has a lot of these things. Now these things are where you can have enzymes binding to them. And what do you call them? You call them accessible sites. Because what are they? They're accessible to enzymes. Accessible, yeah, that's probably not the right terms, but you get the point, don't you? Accessible sites. Glycogen has a lot of these accessible sites. That means more enzymes can break down glycogen at a time. Because they can break down glycogen from so many different places, all that means is that glycogen can be broken down very, very quickly. That in turn means glycogen is a very, very, very good energy store in our body. Now, You might be wondering, yeah, glycogen has a lot of more accessible sites. It can be broken down very quickly, so yeah, it's a good energy store. But why did we need more branches than Amylopectin in the first place? What's different? Well, let's imagine a plant. Plant is here. This plant sits all day in the same place, every day, day in, day out. Whereas us humans, we're walking around. We're taking our muscles, we're contracting them in, down, up, down. We're going berserk. Whereas this guy, he's just there, day in, day out, having a peaceful time. He doesn't need a lot of energy, at least not very quickly. Whereas us humans, we're going, our muscles are contracting hard. We need a lot of energy in a very short amount of time. Plus we need to store that energy too. So in our muscles, we have a lot of glycogen present. And that glycogen is found in our cells, and we need to be able to break down that glycogen very, very quickly. So if I'm at the gym, I'm trying to weight lift, I need to have that glycogen broken down very, very, very quickly into alpha glucose so that I can then use it to pick up the actual weight. That is why glycogen needs to have a lot more accessible sites and therefore needs to be a lot more branched. Okay, so now what I'm going to do is I'm going to write out the actual properties of glycogen that you need to know. Okay, so the first property of glycogen that you need to know is it is insoluble. Now you'll see with all glycogen, amylopectin and amylose, all of these are insoluble. And this is very important, as previously discussed, because it does not affect the water potential. It does not affect water potential. I don't know how many times I've drawn that in, but glycogen does not affect water potential, nor does starch. Number two, more importantly, it is extremely branched. Branched, you can put it in brackets, highly, it's very, very, very branched. That's good for energy release. Very quickly. Why is that important in animals? Because animals move around. Animals move around, they need a lot of energy very quickly. Plants don't move around. Plants don't need energy very, 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 very quickly. Yeah, right, I need a lot of energy right now doing this. So glycogen, these are the two properties. Go off and memorize them. Don't be a fool and not memorize glycogen. Do it. Right, okay. So the final thing I want to move on to is a bit unrelated to this. And that is cellulose. So we've covered starch, we've covered glycogen. The final carbohydrate that you really need to know for your spec is cellulose. And you need to know the structure about cellulose. Now, why cellulose is so important is because it's got a slightly different structure. In the sense, it doesn't necessarily use the alpha glucose molecules we're doing here. It uses beta glucose. Now, beta glucose hasn't been used in any of the previously discussed carbohydrates. No amylose, no amylopectin, no glycogen. All of these are alpha glucose. The only one beta glucose carbohydrate you need to memorize is cellulose. So let's go over that now. Okay, so now what we're going to be talking about is cellulose. So I've drawn out the structure of cellulose here, and you can see that cellulose essentially is just a long chain. That's kind of all that cellulose is. Now, key point, cellulose is made out of beta glucose molecules, not alpha glucose molecules. Okay, to remind you, let's have a look here. So, alpha glucose has the OH group down here at the bottom and has a H up there. Whereas beta glucose is not like that. Beta glucose has the OH instead at the top here. It's OH and this is H. We will come back to these two glucose monomers shortly. But before that, let us do something very important. Let's have a look at the structure. So the structure here, we can see we've got 1, 4. We can see that this carbon here is carbon number 1, this carbon here is carbon number 2. We have a oxygen bridge between them both. In other words, you have a 1,4 glycosidic bond. Now, something very peculiar with this. You see the oxygen here in your ring is at the bottom, whereas the oxygen in the next beta glucose molecule is at the top, and then the next one it's back at the bottom. In other words, these guys are going their merry way. They're one's pointing upwards, one beta glucose molecule is facing that way, and then the other one is kind of like that, so it's facing downwards. And then the next one is facing upwards. In other words, they're alternating. Beta glucose molecules are alternating in the direction they point to. They are vertically alternated. Now, why is this important and why is this happening? Let's have a look. Well, the only way we can figure that out is looking at this diagram. Imagine these both were alpha glucose molecules, okay? Which is what we saw with our starch and glycogen. So we have our OH right here at the bottom and we've got the H up there. Now what you see is this carbon, which is carbon number 1, this carbon, which is carbon number 4. The carbon number 1's OH group is right next to the OH group of carbon number 4. In other words, these two guys can react together and can form the oxygen bridge and the glycosidic bond. In other words, it can easily form a 1,4 glycosidic bond. A 1,4 glycosidic bond can easily be formed between 2 alpha glucose. However, with beta glucose, that is not the case. Oh my god, what's going on? Well, with beta glucose, what you actually see is because your hydrogen is here and your OH is here, your OH is very, 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 very far away from the OH here. So the OH on carbon 1 on this beta glucose molecule is very far from the OH on carbon 4 of the other beta glucose molecule. In other words, you're not going to have a glycosidic bond forming like that. The only way you can have a glycosidic bond forming is if this whole thing turns around. If the whole thing turns around, these two will switch places. In other words, your OH will now be at the top. And if it's at the top, it's right next to the other OH. Voila, you can get a 1,4 glycosidic bond being formed. That is why cellulose has one beta glucose molecule facing upwards, the next one facing downwards. They're vertically alternated. Right, now what's one of the consequences of this? Because it's vertically alternated, that means if you had an angle forming this way, the next angle that would have formed would be this way. So essentially you're forming that. In other words, this is really just straight. So cellulose is one of the very few polymers which is like dead straight like an arrow, right? It's very straight. It's not branched, it's not coiled, it's just straight, okay? Now this is something that's very important. Imagine you're building a bridge, right? When you're building a bridge, you use wood planks, right? Or you might use beams of iron. What do you want all of those beams to be? What shape do you want them to be? You want them to be in a straight line. The same way, cellulose is in a straight line. So what do you think the function of cellulose would be? Yeah, exactly. Cellulose is a sort of structural carbohydrate. You do not come across a lot of these. Remember this one, please. Cellulose is a structural carbohydrate. Cellulose is a structural carbohydrate. Well, now we figured out the small structure, the microscopic structure, whatever. How is this built up? So even when we're building a bridge, we might bundle a lot of beams together and then make a big beam. That's kind of what's happening here. So cellulose, these microfibers, which is what we had a drawing of, is kind of like this. Now these microfibers join up together. When they join up together, these microfibers create macrofibers. So cellulose microfibers microfibers. Microfibers, yeah. Just want to make sure we're doing this right. Yeah, so okay, cellulose has microfibrils. Not microfibers, yeah, microfibrils. These microfibrils join together to make up macrofibrils. Macro means what? Means big, micro means small. Self-explanatory. Right, now the only question that comes is, what's the bonds between these microfibrils? The only bonds that could occur are hydrogen bonds, lovely hydrogen bonds. Hydrogen bonds between microfibrils. Okay, so you have hydrogen bonds between the microfibrils. Microfibrils join up together to create macrofibrils. Macrofibrils are strong. They will support a lot of stuff. That is why cellulose is a very, 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 very good structural carbohydrate. That's why plants like to use cellulose where? Yes, in their cell walls, because they need structural support. Another thing that's important with cellulose is it is insoluble again. Yeah, it's insoluble again, which is again great because you've got water flowing through this. You don't want it to be soluble. Right, so we've kind of covered everything about cellulose too. That it's straight and it's straight chained. It's dead straight like an arrow, so it's good for supporting structures. So it's a structural carbohydrate. Right, so now we've essentially just covered all of the carbohydrates we need to know. We've covered starch, we've covered glycogen, and we've finally now covered cellulose. Starch and glycogen are energy storage carbohydrates, whereas cellulose is a structural carbohydrate. Right, so I hope you guys have enjoyed this lecture and you've learned something about carbohydrates and some examples of them. Please keep learning and please stay curious and keep watching Newtonian. Thank you.